Okay, so for the last few weeks, the genetic disorder that I've been researching is the sickle cell disease. So just a little bit of the history of the disease. It was thought to have originated in Africa several hundred years after it was actually discovered in America in the 1900s. In 1910, Dr. James Herrick treats a patient with anemia and he looks at the red blood cells under a microscope and coins the term sickled. In the 1930s, it is suggested that pain associated with this disease is from the clogging of sickled cells in blood vessels. In 1949, Dr. Linus Pauling discovers that sickle cell disease is caused by an abnormal hemoglobin. In 1957, Dr. Vernon Ingram discovers specific amino acid changes that causes the sickling. And then if you'll notice, there's kind of a gap from 1957 to my next point in the 1990s. In between this time, they kind of are just trying to do more research on the disease and figure out more about it, and then different tests that can determine whether a person has sickle cell disease. There's not really any big specific discoveries between this time. But in the 1990s, hydroxyurea and bone marrow transplants are used as treatments, and then they figured out that bone marrow transplants can also eventually cure the disease. And then 2000 to 2010, gene therapy and stem cell transplants are studied as cures. So some of the physical effects, obviously it causes the sickle-shaped red blood cells. This change in shape can cause blockage of blood flow in blood vessels, which in turn causes tissue damage to the lungs, brain, kidneys, and spleen. This tissue damage can also cause pulmonary hypertension, stroke, acute chest syndrome, and eventually sometimes death. It also causes a low number of functioning red blood cells, so this causes anemia, infections, and pain. And anemia can cause shortness of breath, fatigue, and a delayed development in children. And this is a picture that I actually got from Wikipedia, but I found out that it kind of illustrates what sickle cell is really well. So in A, you have like the normal red blood cells. They're kind of a donut shaped and they're more flexible, so they can flow easily through the blood vessels. And then right here you have kind of what a normal hemoglobin looks like in this cross section of red blood cells. And then in B, you have the abnormal sickled red blood cells. So they're kind of like a banana shaped. And so right here, like you can tell that they're blocking the blood flow in red blood cells and in the blood vessels. And then here is the cross section of the sickled cell. And you can tell that the abnormal hemoglobin is kind of elongated, kind of gives that sickled cell appearance. So the prevalence of this disease is at least 100,000 Americans are affected, and a 1 in 12 and African Americans and 1 in 100 Hispanic Americans are carriers. So the molecular cause is caused by a mutation in the gene hemoglobin beta subunit, which is located in chromosome 11 in humans. So hemoglobin basically has 2 alpha and 2 beta subunits. The mutation causes a change in the amino acid sequence of beta globin, and this causes an abnormal hemoglobin called hemoglobin S. There's several different types of hemoglobin. Hemoglobin S replaces one of those beta subunits. And then there's actually the specific change in amino acid is from glutamic acid to valine in position 6. and this causes the side chains to stick together. And so this kind of illustrates what I just talked about. Here you have the normal protein. There's a mutation in the DNA, and this changes the protein to valine from glutamic acid, and there's that. And then here's what normal hemoglobin would look like, and then here's the clumped hemoglobin. As you can tell, it kind of has like the sickle cell shape almost, and obviously, like if you had the clumped hemoglobin going through your blood vessels, that is going to cause some blockage compared to like this normal that's right here. So the mode of inheritance is autosomal recessive. This basically means that both parents must have at least one recessive allele. Both parents can be carriers, or one parent can be a carrier, and the other parent can be affected, or both parents can be affected. There just needs to be at least one recessive allele from both parents. And then this is the pedigree that I created. So you'll see right here, Steve and Gladys, they're both carriers, and they had one child that was affected. Since it's recessive, it's little a. And David married Julie, and she was a carrier, so they had 2 children that were affected. And then Chris right here, he is autosomal recessive, had children with a carrier, and they also had 2 children that were affected. So some of the genetic testing is that all newborns in all 50 states, they're all screened for sickle cell. That's one of the diseases that are checked off for whenever they do newborn screening. Some of the other testing includes hemoglobin electrophoresis, which measures the amount of each type of hemoglobin. Like I said, there's multiple different types of hemoglobin, so this can show how much of one specific type that a person may have. There's also DNA testing and mapping, and this can help determine if a person has the associated mutation with sickle cell, like the previous mutation that we discussed, from glutamic acid to valine. High performance liquid chromatography, and this is basically used to identify substances or compounds in a mixture, for example, like blood. And then finally, one other way is through amniocentesis. So this is something that they might recommend if a pregnant woman is a carrier for the disease. That way they can test the amniotic fluid and see if that child is going to have either be a carrier or be affected with the disease. And then finally, here are my references, and that's it.